I think many of us can relate to the anxiety and uncertainty surrounding getting the diagnosis and starting treatments. We're grateful for your insights and inspiration. We also want to thank Dr. Kotaya for her excellent NETS 101 presentation that gives us a good overview of NETS. Our next speakers are on the topic of clinical trials. As those living with or supporting those living with NETs, it's important for us to know how to interpret research articles and understand clinical trials that we might be looking into. It helps to understand how our NET experts weigh the clinical studies in making our treatment decisions. We will first hear from NET patient Mary, who shares about her own experience in a clinical trial that changed her life. Next is NET oncologist Dr. Dan Lee, co-director of the Neuroendocrine Tumor Program at City of Hope in Duarte, California. Dr. Lee leads several clinical trials on NETs, and in fact, he recently gave 2 oral presentations at the Professional Medical NET conference called NANETS, which stands for the North American NET Society. One of these presentations was on NET Vitals, which you may be familiar with because it was created by our late founder, Giovanna Mbesi, along with myself and Dr. Dan Lee, with the goal of improving patient-physician communication. You can find the NET Vitals tool and a webinar to help you complete it on our website. Dr. Lee will be giving a presentation titled Clinical Trials 101 to explain research terminology and concepts. He will go over 2 examples of groundbreaking studies that are the cornerstone of our treatments with somatostatin analogs, sandostatin and lanreotide. After Dr. Lee's presentation, NET endocrinologist and oncologist Dr. Heidi Del Rivero from the National Cancer Institute, NIH, will present on NET clinical research. She will describe some of the current and ongoing NET studies that are open for enrollment. We will now hear from Mary, followed by Dr. Lee, and then Dr. Del Rivero.
I'm pleased to have the opportunity to talk to you on the topic of clinical trials, as I have spent a number of years in my cancer journey on a clinical trial. In 2005, I was diagnosed with a pancreatic NET and underwent the Whipple procedure. I had no evidence of disease for a number of years, but eventually had growth in my liver and was in need of a systemic treatment as my liver lesions were considered inoperable at this point. I was fortunate enough to be enrolled in a trial at Cedars-Sinai by my oncologist, Dr. Wolin. My husband and I had extensive discussions with my doctor about the risks and the benefits of the trial and other treatment options, and we decided this was the best choice for me. I will be honest when I say that I was very scared about this treatment due to the fact that it had not been approved for pancreatic NETs at that point. There were 2 separate arms of the trial, and my arm was sandostatin injections along with everolimus or Afinitor, which is an oral chemo. I joined the trial in 2012, and 5 years later, I received an email from my doctor with the subject line, quote, "You beat your study to submission," unquote. He went on to say, "Congratulations for being the last patient and single-handedly keeping it open several years longer than expected." The clinical trial was a success and became an accepted treatment modality for NET soon after. And I'm proud to say that 8 years after my enrollment in the trial, I'm still on the same treatment protocol and continue to live a very fulfilling and active life with stable liver metastases. Many people think of trials as a last-ditch effort, but that's absolutely not the case. The way to think of a trial is more that it can be a viable option, a treatment option at any point during your cancer journey, from very early on in your treatment, possibly after a surgical intervention, or even when you're at a decision-making point later in your diagnosis. I cannot stress enough the importance of being under the care of a NET provider if you have any interest in being enrolled in a trial. A non-specialist will not be as informed of the many trials that are available, and most importantly, the one that is right for your specific disease. The zebra aspect that we're all familiar with NET cancer makes it much more complicated to determine the best course of treatment. So having a NET specialist is critical. It's also really important to note that with a rare disease like ours, it's much more challenging for researchers to find enough patients to fill the studies. So any assistance that any of us can provide will benefit our entire community. I'm often asked, am I glad that I did it? And if what I would tell other patients about being in a trial. I can't stress how much I valued being a part of my trial. Obviously, high on the list of reasons is that the treatment has been a huge success for me. I also felt that the trial was really well managed, and I felt extremely well cared for by my team. There were very tight protocols on the labs and the scanning and the distribution of medication. I had special treatment room assignments, and the list goes on. But all this made you feel like you were really well cared for. I had a study coordinator that monitored me every step of the way, which was very comforting. I'm so grateful for my clinical trial and that it has afforded me years of stable disease. And equally gratifying is that many other NET patients will benefit from the trial as well. 15 years later, I've been witness to multiple medical advancements in the NET world, thanks to other clinical trials that have occurred since my original diagnosis. This is proof that our involvement in trials has the potential to make a huge impact on expanding our treatment options and moving the field forward, which clearly benefits us as well as all of those coming after us. Thank you so much.
Great. Good morning, everyone. I'm Dan Lee. I'm a GI medical oncologist at City of Hope, and I co-direct the Neuroendocrine tumor program. So today, I'm going to be discussing with you understanding clinical trial terminology, NET patient basics. And I think what I want you to get out of this presentation is really some of the key fundamental basics in terms of how we read and understand some of the clinical trial data. And I think that's absolutely vital to NET patients, because if you understand the data that you're reading, then you will have much better knowledge and insight to some of the treatments that might be coming that are able to help you. So in this presentation, I'll be going over a couple different things. First, we'll highlight the general research process that's involved in doing clinical trials. Next, we'll highlight some of the statistical terminology that are used when you're trying to read and understand these clinical trials a little bit better. And then finally, we'll look at a lot of these clinical trials. When they're designed, they have certain types of outcomes that they're trying to address. And these are the really kind of the primary objectives of these various clinical trials, so that you have a better understanding of what those trials are trying to accomplish. All right, so we'll start with initially with the drug development process. I think this is very important to recognize, because oftentimes it's very difficult to get an understanding of how much work it goes into in terms of a medication that ultimately gets FDA approved for neuroendocrine tumors. So initially with the drug development process, in terms of drug discovery, we usually will start with approximately 10,000 different types of compounds. And from this, only about 250 compounds ultimately make it to what's known as preclinical work. So this is usually work that involves animal testing, as well as higher primate testing, before they can be considered for human clinical trials. And out of those 250 compounds, then ultimately 5 compounds make it to actual clinical trials. And these are trials where we're testing them in humans. And then out of these, usually only 1 will ultimately be FDA approved. Now, going through this whole process, what you can see is that it takes about a little over 6.5 years from initially screening those 10,000 compounds and then ultimately leading to 1 FDA approved treatment. So there are really 4 major phases of a clinical trial. Initially, all trials will start with what's known as a phase 1 trial, which is really focused on the safety, as well as trying to establish a safe dose. So usually in a phase 1 trial, you're really assessing for toxicity from the treatment. You're also looking at how the drug or medication is ultimately absorbed and distributed throughout your entire body. Initially, you're not going to necessarily find too much about whether or not the drug truly works or not, but you can sometimes get a hint of it. And the trials are getting more sophisticated over time. So sometimes after you establish initial safety, you might see what's known as a phase 1b extension trial. This is where once they develop an initial safety signal from the medication, it will then expand to a group of patients to see whether or not the drug is efficacious. But then traditionally, efficacy is really evaluated in what's known as the phase 2 clinical trial setting. This is where we already have a safety signal from the phase 1 clinical trial. And now we want to test the drug to see whether or not it actually works for the tumor type that we're treating. In addition, a phase 2 trial also helps to develop additional short-term side effects as well as safety, in addition to the initial phase 1 trial that it went through. And if a drug is found to be safe through a phase 1 trial, and that safety is confirmed in a phase 2 trial, and in the phase 2 trial is shown that the drug is efficacious, then the drug usually will then move on to what's known as a phase 3 trial. In the phase 3 trial, usually the newer drug, the experimental treatment, will then be compared to a current standard of care. So kind of an established treatment for neuroendocrine tumors. Now, sometimes you might see a phase 3 trial that's randomized to what's known as placebo, meaning that it's essentially like a sugar pill if it's an oral medication. Sometimes that's done because, let's say it's a trial that is testing a medication for neuroendocrine patients after they have gone through all the different FDA-approved treatments. So the current standard would be no additional treatment. So this is where kind of a placebo is potentially justified. But that's something to be aware of whenever you're participating in trials, because if you're not comfortable being randomized to the placebo control arm, that's something to consider. And a lot of these trials that are phase 3 that's randomized to placebo might have something that's known as crossover. This is where you might start on the control arm with placebo, but if you develop disease growth on that arm, the trial will allow you to cross over and participate in the experimental treatment moving forward. So that's something else to kind of keep in mind. And then finally, once a drug is FDA approved, there's what's known as a phase 4 trial, which is essentially a post-marketing study to obtain additional long-term safety and as well as efficacy of the medication in the real-world setting. So now that we have understood kind of the different phases of the drug development process as well as the clinical phases of a clinical trial, the next important thing to understand about clinical research, and especially when you're reading about the data from these trials, is some statistical concepts and terminology. So we'll start with what's known as mean and median. So mean is essentially the average. So oftentimes you'll see a clinical trial that presents a table of different types of patients that were included in the trial. And it might include the mean age of the patient, so the average age of the patients that participated in the trial. Usually, I would say mean is not really used these days in a lot of the reporting of clinical trials. One of the reasons for that is that oftentimes there's wide variation of the mean in the trials. So this is where the median really comes in. So the median is essentially the middle value in a set of numbers. So it's often used when 50% of patients experience an event. So you could have a table of participants where it lists that the median age of participants participating in that clinical trial might be 60 for neuroendocrine tumors. And then it might also report that the median Ki-67 proliferation index, which is a way to grade your tumor, might be, let's say, 5%. Okay, and then it might also report a outcome of the trial, such as how long patients lived in terms of survival, and then it will usually report it as a median survival, meaning that what is that middle number for how long patients lived with that treatment. In addition to mean and median, something to really understand is something that's known as the hazard ratio, or HR, and you'll see this, and I'll give you some examples. So this is what the hazard ratio refers to, is the relative risk of an event happening in one trial arm versus another. And this is really important because a lot of your key outcomes will include this hazard ratio. What you'll see is that if the hazard ratio is 1, it means that there is absolutely no difference between the treatment groups, whether it's the experimental treatment or the standard of care treatment. If the hazard ratio is 2, essentially this is a doubling of risk of the event. And usually if you're talking about survival, you don't want that hazard ratio to be above 1. That means the experimental drug is actually doing worse than the current standard of care drug. And usually you want the hazard ratio to be less than 1. So for example, if the hazard ratio is 0.5, it means that it would be half the risk. So if we take survival, for example, and you see a clinical trial that reports that the hazard ratio for overall survival of the experimental treatment to the current standard of care treatment is reported as 0.60. What this means is that there's a 40% reduction in the risk of death with the experimental treatment. So that's good. So anything less than 1 is going to favor that experimental treatment. And you would take 1 minus whatever hazard ratio is to get the percentage in terms of the percentage reduction in the risk of death. And this hazard ratio is really important because it accounts for differences in events over the entire period of clinical trials, and that's why it's used this way. Finally, one of the key things that you'll see in a lot of clinical research is something called the p-value. So this is the probability of obtaining a statistical result by chance. So this is what is usually reported in a clinical trial if it is statistically significant or not. The lower the value, the lower the p-value, the less likely that the findings from the clinical trial was a result of chance alone. And that's important. It's actually then really driven by the experimental treatment, for example. The typical threshold that we set for the p-value is 0.05, and each trial will have a different threshold for the p-value. What this means is that only 5% probability of the final result from the clinical trial actually happened by chance, so that you're most likely seeing that the events that happened in the clinical trial was actually due to the actual treatment itself and not by chance alone. As I mentioned, there's different types of p-value thresholds for different types of trials, and sometimes the p-value might be a little bit lower. So just because a p-value might be less than 0.05 for one trial, it might not be necessarily, and it might be significant for that trial. It might not necessarily be significant for another trial, because the other trial might have a lower threshold when they were designing the trial to achieve for a p-value, such as 0.025, for example, to achieve statistical significance. So in addition to the p-value, something else that you can use to see whether or not something is significant and also whether or not something is very precise is something that's known as the confidence interval. So this is oftentimes something that will also be reported. So the confidence interval is essentially the percentage of certainty a result will fall within a given range. So as we mentioned, oftentimes a hazard ratio will be reported. And if it's less than 1, it's good, meaning that the experimental treatment is potentially benefiting. So the hazard ratio will also be reported as a 95% confidence interval. And it indicates that 95% certainty that the hazard ratio will fall in a certain range. So for example, if the hazard ratio was, let's say, 0.55, and the 95% confidence interval was 0.4 to 0.7, what it means is that you have 95% certainty that this hazard ratio will fall between 0.4 to 0.7, indicating that clearly the treatment works. Now, if you have this confidence interval that's very narrow, let's say instead of 0.4 to 0.7, it's maybe 0.5 to 0.6, what that tells you is that you really have a lot of confidence in what the result is showing, and you truly know the significant benefit of the treatment, and it allows you to have better precision from those results. This is important because you clearly don't want a treatment that, let's say, has a 95% confidence interval of a hazard ratio that, let's say, ranges from 0.4 all the way up to 10, for example. So here it crosses 1, which is our threshold of equivalence in terms of potentially the experimental treatment to the other treatments, the standard of care treatment. So if you have also a large range that crosses that, you really might not know whether or not the treatment is actually working compared to the standard of care. Once you understand kind of the different phases of the trials, and you have kind of these basics in terms of statistical terminology, the final thing that you want to look for is what were the key kind of clinical trial endpoints for all of these trials. So usually the primary endpoint for many studies in oncology will be something that's known as overall survival. This is defined as the percentage of patients living at a defined period of time. This is traditionally the absolute gold standard for oncology clinical trials, and it's often reported as a 5-year survival rate. However, overall survival is very difficult for neuroendocrine tumor research. The reason why is because oftentimes for neuroendocrine tumor patients, as you know, with a more indolent disease, patients might live for a long period of time. In addition, they're going to be treated with numerous different types of treatment. They might start with somatostatin analogs, they might go to a target therapy, they might go and be treated with PRRT. So their overall survival is actually influenced by multiple treatment and not just one treatment that they get. So in this setting, it makes overall survival for neuroendocrine tumor research very difficult to achieve because someone's overall survival is not just dependent on that line of therapy that they're receiving at this one period of time, but it can be influenced by later treatments as well. How do we try to counter this in neuroendocrine tumor research? Well, what we oftentimes do is then we focus the primary endpoint to be what's known as progression-free survival. So progression-free survival just refers to the percentage of patients who continue to live with a disease that is stable or better. The reason why this is an important objective measure is that we know that when patients and their tumors start to grow, that progression is often symptomatic. And this progression-free survival endpoint is not affected by crossover, meaning that if, let's say, a patient starts on an experimental treatment and they got into the control arm, and then they crossed over to the experimental treatment, your progression-free survival did not change, right? So your progression-free survival is the time when you first developed confirmation on a scan that your disease is growing, even on the control arm. And then when you cross over to, let's say, the experimental treatment, then it resets and you try to define what was the time until you develop progression on the experimental arm. So this is a nice endpoint, and it's not also impacted by, let's say, later lines of treatment, because once you have confirmed that your disease is growing, then you know how long the current treatment was effective for you. And it's not like overall survival that's influenced by additional treatments that you may have later on. In addition to progression-free survival, another key endpoint that we look at in neuroendocrine tumor trials oftentimes is quality of life, because we not only want to know whether or not the treatment is effective, but we also want to know whether or not your quality of life is negatively impacted or whether or not it's actually helping your quality of life. And this is really can be done with various different types of questionnaires and scales. And there's different ways to look at quality of life. You can see whether or not your quality of life will improve with treatment. But oftentimes what we look at is whether or not there's a deterioration in your quality of life over time on the experimental treatment that you're on compared to the standard of care treatment, to get a sense of, potentially how negatively a new treatment might be impacting your quality of life. And that's really good to know, because we not only want the treatment to be working for you, but we also want to make sure that your quality of life is preserved as well. So let's look at some examples, since we've gone through a lot of terminology in terms of how we look at some of these clinical trials of FDA-approved treatments for neuroendocrine tumors, and begin to get a sense of how we interpret some of the results and understand whether or not the treatment is effective for us. So the PROMIT study was one of the first studies for neuroendocrine tumors, and it really was what's known as a randomized study. It was a placebo-controlled, double-blind randomized study, meaning that patients were randomized to receive either octreotide long-acting injection versus a placebo injection to see whether or not there was control for tumor growth in patients with metastatic mid-gut neuroendocrine tumors, so really neuroendocrine tumors kind of throughout the small intestine. Back then, when the PROMIT study was reported, this just shows you kind of the patient demographics of the patients within the study. You'll see that there's equal number of patients essentially in each arm, as you would expect for a randomized placebo-controlled study. So there were essentially half of the patients in the octreotide long-acting injection arm, and then half the patients in the placebo arm. They will report kind of this median age in terms of what was essentially the middle age for patients that participated in the study. And that was relatively consistent with those that had a diagnosis of neuroendocrine tumor. They will then report kind of the median time to diagnosis, and then a lot of different types of tumor characteristics, where they will give you the number of patients as well as percentage of patients, so that you can understand what were the patients that were enrolled on this clinical trial specifically. And then all the way to the right side of this table, you'll see this p-value on the side. And for a p-value for a baseline characteristics table, what you're trying to see is that the p-value is not too low, essentially less than 0.05. The reason why is you want the arms to be balanced. So you actually want them to be not any different to each arm. So remember, the p-value just tells you whether or not something occurs by chance or not and everything. So if you have a really high p-value, what it means is that there's no significant difference between the two treatment arms, which is exactly what you want for baseline characteristics. So this is kind of the opposite of what you want for, let's say, outcomes comparing the experimental treatment to the standard treatment. You just don't want to see any differences between the patient characteristics between the two treatment groups. And this was fairly consistent in this trial. One of the ways that clinical trials will often present a key outcome, especially related to, let's say, overall survival or progression-free survival, or even quality of life, is that they might use something that's known as a Kaplan-Meier curve. What this curve tells you is essentially the proportion of study population still surviving or free of disease, or hasn't necessarily progressed on the current treatment at successful times. It is really the common method to represent overall survival or progression-free survival or efficacy. And the larger difference that you see between the curves, between the experimental treatment versus the standard of care treatment or placebo treatment, it just shows you and it tells you the greater difference in the treatment arms. So this is the PROMIT trial again, and this is how the Kaplan-Meier curves are presented. So on the top of this figure, in terms of the A part of the figure, this is essentially the analysis focused on progression of the tumor between those patients that were treated with octreotide long-acting injection versus placebo. And what you see is that there's a great separation of the arms in terms of time to progression, favoring treatment with octreotide compared to placebo. So the octreotide is shown in yellow, the placebo is shown in blue. And what you also see a little bit further down in the kind of the footing of the A panel is that you see a p-value And then you also see a hazard ratio. Again, the p-value is clearly less than 0.05, meaning that the results are statistically significant. And then what you see is that the hazard ratio is 0.34. What this means is that there's essentially a 66% reduction in the risk of progression or death if you're treated with octreotide long-acting injection versus placebo. And the data also reports this 95% confidence interval, which is also relatively narrow. So it shows you that the results are also very precise. So this is exactly what you would want for a treatment that's being tested for a patient with neuroendocrine tumor. You're seeing that the treatment is effective compared to placebo in terms of tumor control. Now, in panel B, what you'll see is the overall survival data for those patients that got treated with octreotide long-acting versus placebo. And now what you can see is that the Kaplan-Meier curve does not separate. They're essentially on top of each other, meaning that overall survival was very similar between the group that was treated with octreotide versus the group that was treated with placebo, which makes sense, right? Because many of these patients might have come off trial for placebo. They might have gotten additional somatostatin analogs further or additional other treatments, just like the experimental treatment option. So there's no difference that's detected in terms of overall survival. And as I mentioned, that's because it's influenced by additional treatment after the clinical trial was done. And that's why oftentimes for neuroendocrine tumor trials, you might not see that overall survival difference. In addition to this, there's kind of, subgroup analysis that's usually done in a clinical trial. So they'll try to see, who are patients that are included in that trial that really benefit from treatment. In this PROMIT trial, they looked at, did patients that have carcinoid syndrome or essentially functional tumors, did they benefit? Did patients that have a lot of liver burden, the percentage of liver involvement, did they benefit? Did patients that were younger versus older, did they benefit? Did patients that have primary tumor resection, did they benefit or not? So what you can see here is that, again, this hazard ratio number comes on the right side of the figure, and it's good. It's all clearly under 1, and that's beneficial. And then if you look closer in terms of the 95% confidence interval, you'll see that some will have a narrow range where it's clearly under 1, but then some will potentially cross 1. So for example, liver involvement 10 to 50%, your 95% confidence interval is 0.10 to 1.67. So that's a wide range. And you don't know truly how precise the data is for someone that has significant liver involvement. Now, I will say you can't say that it doesn't work either, because it could just be that your patient size is relatively small, and as a result of that, you have this wide range. So if you look at the number of patients that had 10 to 50% liver involvement, it was only 14 patients, whereas you definitely had more patients that only had 0 to 10% liver involvement, 52 patients. That's where your hazard ratio is more narrow. So these are kind of the little nuances that you have to kind of pick up. But in general, for subgroup analysis, this is really exploratory, and you want to just get a general sense that overall the hazard ratio is benefiting for all different populations. If you see that there's one population where it's not benefiting and the hazard ratio is very high, meaning that the experimental treatment, it might not be working, and the confidence interval is very, very wide, even when you have a large number of patients, then you would be a little bit suspicious of whether or not in that subpopulation, whether or not the treatment is working compared to the overall population. Let's look at the lanreotide trial. That was also a phase 3 randomized double-blind placebo-controlled trial comparing lanreotide, which is the other somatostatin analog, versus placebo in patients with well-differentiated or moderately differentiated non-functional somatostatin receptor positive gastroenteropancreatic neuroendocrine tumor. Primary outcome of this study was progression-free survival, as we mentioned, because overall survival is very hard to demonstrate for an initial first-line trial. The Kaplan-Meier curve for progression-free survival separates very clearly on this. So it met its primary endpoint. The p-value was less than 0.001, so that's good. That means the results were significant. The hazard ratio was 0.47, meaning that there was essentially a 53% reduction in the risk of progression for those patients that were treated with lanreotide versus placebo. And you have a narrow 95% confidence interval, meaning that the results are fairly precise. So this is usually how researchers will present their data for the different subgroups that's involved in a study. There's some things to kind of keep in mind for a forest plot. The size of squares is usually proportional to the importance or the weight of the data, meaning that if there's more patients involved in a study and they are looking at these various different subgroups, if the sometimes the square that's larger means that there's potentially more weight put into that subgroup, usually because of more patients. The horizontal lines essentially represent the confidence intervals that we talked about. So it's a different way, it's more of a pictorial way to represent the data. So here I show you a forest plot from the lanreotide study. Again, these are looking at kind of subgroup analysis. So you already saw the data in terms of how the treatment benefits versus placebo for all comers. But then this shows you, and it looks like looks at the subgroups in terms of breakdown. And what you will see again is that generally on this subgroup analysis forest plot, that everything favors lanreotide with a relatively narrow confidence interval. The only thing that you don't see is for those patients that have high gut tumors. So these are patients that might have tumors of the neuroendocrine tumors of the colon or the rectum, where you have a large range now in terms of your 95% confidence interval and your hazard ratio is above 1. So you can't truly say that lanreotide is necessarily favoring this population. But then what we also know is that the number of patients, again, in this group was only 14 patients. So you can't say that truly that lanreotide is also not benefiting these patients. You just have a much wider range now because you only have a small number of patients to really get that precision that you would want, which is what the 95% confidence interval is trying to tell you. In addition to the Kaplan-Meier curves as well as the forest plot, another thing that you will see as a clinical trial outcome is what's known as response to therapy. And this is essentially categorized as response rate, or, are my tumors essentially shrinking or not? So response rate is really based off of a tumor size measurement. And in clinical research, we often characterize it based off of something that's known as the RECIST criteria, which is just a fancy medical term for response evaluation criteria in solid tumors. It's our criteria that we use as researchers to classify whether or not a treatment is responding to treatment or not. So what you'll see is that it's not necessarily just any tumor size shrinkage. If a patient has a complete response, meaning that essentially on a scan, they have no evidence of measurable tumors after they start treatment. A partial response means that you have had a greater than or equal to 30% reduction in the size of all measurable tumors. So usually when you start research, we'll pick certain type of tumors that we look at from the very beginning. And if there's overall a greater than or equal to 30% reduction in the size of these tumors over time on the clinical trial treatment, then we will say that you've had a partial response. Stable disease, meaning that you have not either achieved a partial response or you have not achieved what's known as progressive disease, which I will show you now. And progressive disease is really defined as greater than or equal to 20% increase in the size of the tumors. So as you can see, this is not what you traditionally will think about necessarily clinically, but this is really what you see in terms of research, because you could, let's say, have 10% growth of your tumors and still be called stable disease because you did not meet the requirement for progressive disease based off of this criteria, and you did not meet the criteria of partial response. But we know that probably your tumors are growing, and clinically, probably you are progressing, even though by research, you don't meet that. So this is something to tease out on the clinical trials. Similarly to that, if your tumors are shrinking, let's say by 10%, it's still called stable disease, even though you're probably responding to treatment because you're having some tumor shrinkage from the treatment. It just doesn't meet the threshold of what's classified as response. So when you ask your doctor what's the response rate of a particular treatment, and they tell you that the response rate is overall 10%, that doesn't mean necessarily that it's not benefiting a majority of the patients. It just means that only 10% of the patients had that greater than or equal to 30% reduction in the size of all measurable tumors to classify as a partial response. So this further clarifies this. So oftentimes you will see a report for the objective response rate, which is a percentage of patients that had a partial response or complete response with treatment. You'll also sometimes see that clinical trials will report a disease control rate, which is the sum of a complete response, partial response, or stable disease rates. And then there's also a duration response, so how long patients maintain their partial response, complete response, or stable disease in total. So response is usually presented at a clinical trial with something that's known as a waterfall plot. So this reflects individual patient response rate to the treatment itself. So this is the RADIANT-3 trial that looked at a medication called everolimus for patients with advanced pancreatic neuroendocrine tumors. And this is the waterfall plot that was presented. So what you can see is that if you just compare, let's say, response rate per RECIST criteria, the objective response rate for everolimus was only 5% versus placebo. So you might, as a patient, say, oh, this is not a good treatment because there's only 5% response rate. But again, that's 5% of those patients that had 30% or more shrinkage of the tumor. For those patients that got treated with everolimus, shaded in the purple, is all those patients that had shrinkage of the tumor to some extent. They might not have met the 30% to classify as a response, but they still had shrinkage of the tumor. And if you look at the table in terms of decrease in the size of the target lesions from baseline, what you'll see is that 64.4% of the patients that received everolimus had decrease in the size of the target lesions, compared to only 20.6% of patients that were on placebo. So this is the waterfall plot for sunitinib. And as you can see, a majority of patients had shrinkage of the tumors, but only 9% of patients actually met that threshold of 30% or more to classify as an objective response rate. So those patients that had complete responses or partial responses. But again, this waterfall plot informs you much more of those patients that had, responses compared to no responses. And that's why you see that a lot of these patients are now falling into this stable disease category of 63% with a sunitinib arm. But that 63% are actually those patients that are shrinking in size of the tumors. And you say, hey, well, the placebo arm also had 60% of patients that had stable disease. Well, if you look at the waterfall plot, that's because a majority of patients, they had some growth of their tumors, but it just didn't meet the criteria for progression of disease of 20% or more growth. So they were still classified as a stable disease. So when you look at these tables, sometimes they can be misleading, and you might get more understanding of the efficacy of the treatment in terms of shrinkage of the tumors or stabilization by looking at the waterfall plot. Finally, after you understand kind of the efficacy endpoints, there's always safety or toxicity to treatment. And this is usually reported as adverse events. So adverse event is really defined as an unfavorable symptom, sign, or disease associated with use of treatment. And there's different levels of grading. A grade 1 adverse event to the experimental treatment or the standard treatment is essentially mild or no symptoms, and treatment is not indicated. So you don't do any additional treatments. A grade 2 adverse event is essentially moderate, meaning that non-invasive intervention may be indicated. So if you, for example, develop nausea or vomiting, you might want to intervene with anti-nausea medications. A grade 3 event is that it's severe, but not immediately life-threatening, but oftentimes a grade 3 event is characterized by a hospitalization or going to the emergency room. A grade 4 event is what's known as a life-threatening consequence and urgent intervention is required. So these are patients that are definitely hospitalized and they're really sick, and sometimes they might need to be in the ICU. And a grade 5 event is that, unfortunately, the patient passed away from the treatment itself. And that's obviously very serious. So if we look at another clinical trial for neuroendocrine tumors, now the RADIANT-4 trial, which also was a phase 3 trial looking at comparing everolimus versus placebo in patients with well-differentiated non-functional neuroendocrine tumors outside the pancreas, so of the GI as well as the lung, we can really kind of look at these adverse events. So usually there will be a table in the clinical trial showing you all the different adverse events. And this will really allow you to compare the experimental treatment, in this case, everolimus, versus the standard of care treatment, in this case, placebo. And you can really compare the two treatments to see, what is the percentage of patients that develop these various different types of toxicities, whether it's fatigue, diarrhea, rash, nausea here, and it's all included here. And that's a way for you to get a sense of, potentially, what are the side effects that can be encountered with this treatment, and potentially how toxic the treatment is. Usually, we want to really focus on, kind of serious events like grade 3 or higher events, because obviously those are the ones that are potentially not being able to be managed outside of the hospital. So kind of keep in mind and highlight that area, particularly in terms of safety. So in terms of approaching a clinical trial, I think what you need to ask your doctor is, what is the phase of the study? Is it a phase 1 study that's looking at essentially safety? Is it a phase 2 study that's already looking at efficacy and trying to detect efficacy? Is it a phase 3 study where now it's a randomized study comparing the experimental treatment to something else, and that something else could be placebo, and you need to know that? Or is it a phase 4 study, essentially a post-marketing study? So you need to join the study that you're most comfortable with. The advantages of phase 1 and phase 2 studies is oftentimes that there's no randomization, so you automatically get the experimental treatment. The disadvantage is that you have less data initially. You only have preliminary data in terms of safety and efficacy. And this is really where phase 3 comes in, where you have already that safety data and initial preliminary efficacy, but oftentimes it's a randomized study, so you have to be okay with the randomization. What is the primary endpoint of the study? So is the primary endpoint response in terms of significant proportion of patients that are getting significant tumor shrinkage? Is the primary endpoint progression-free survival, how long you can have disease control before you progress? Or is the primary endpoint overall survival? You want to get some of those details to understand you want. Like I said, earlier phase studies will initially focus on response. And then later studies like phase 3 studies and neuroendocrine tumors will probably focus on that progression-free survival endpoint. What is the treatment and control arms? So you want to know what is the experimental treatment and how comfortable you are in terms of understanding that treatment and what preliminary data your physician might know about that treatment. And what's the control arm? Is the control arm a current standard of care, another drug, or is the control arm placebo? And you have to be comfortable with that. And along with that, does the trial have crossover, meaning that if you're randomized to the placebo arm, for example, are you able to cross over to the experimental arm? What is the patient population? So you want to look at essentially, once a trial is reported, what were the patients that are included in the trial? And if you're participating in a trial, you want to know, well, what is the patient population? Is it including all neuroendocrine tumors, including lung, pancreas, as well as GI neuroendocrine tumors? Or if you're a pancreatic neuroendocrine tumor patient, is it specifically a trial just for pancreatic neuroendocrine tumors? Because you might want to be more specific on the trial that you want to choose. How are the results presented? So once a trial releases its results, you want to see all those different presentations that I talked to you about in terms of the Kaplan-Meier curve, focusing on survival, in terms of the waterfall plots, looking at response and tumor shrinkage, and then forest plots looking at kind of subgroup analysis in terms of the patient population that was treated. And are the results significant? So you want to look at the p-value and then the 95% confidence interval to see if it's really precise, and also the hazard ratio to see if the experimental treatment really benefited, and are the results statistically significant. And what are the toxicities to treatment? So really looking at that table of adverse events to see, how patients tolerated treatment and what are the common side effects that patients might have reported in clinical trial, so that when you go on that treatment, what to expect. So with that, I want to thank you for, attending this session and everything. And hopefully I've been able to educate you a little bit on the basics of clinical trial terminology and how you should kind of look at clinical trial data. So when you're thinking about all these different types of treatments, you can make a wise as well as knowledgeable decision, and also a knowledgeable decision in terms of participation on clinical trials. Thank you so much.
Hi, my name is Jaydira de Rivero. I am an assistant research physician at the National Cancer Institute. Today it's my pleasure to talk to you about how can we optimize neuroendocrine tumor clinical research. I know that you have been having a lot of discussions throughout the day about the different classification of neuroendocrine tumors, as well as the different anatomic distribution of neuroendocrine tumors. But this is just as a reminder of the different types of neuroendocrine tumors, which can be localized anywhere in the body. We can see here on the green circles that we can have neuroendocrine tumors in the lungs, neuroendocrine tumors in the pancreas, of the GI tract, of the small intestine. There is also a smaller cohort of patients of neuroendocrine tumors that is localized in the adrenal gland, and we're going to talk a little bit about that, since pheochromocytoma, which is a neuroendocrine tumor of the adrenal gland, is also considered an orphan disease and with not that many treatment options. And we're going to discuss what are the opportunities that we have available at the NIH. Something important to know about neuroendocrine tumors is that neuroendocrine tumors is a very important group of neoplasm that has very unique tumor biology. And at the same time, there is very challenges in terms of deciding the best treatment algorithm, especially in the advanced setting. This is just as a reminder that the neuroendocrine tumors has also increased exponentially over the last few decades. We can see the frequency as well as the incidence of neuroendocrine tumors in the pediatric population as well as an adult population. We have seen more and more patients in the pediatric population that present in my clinic with neuroendocrine tumors in different locations in the body. Pancreas neuroendocrine tumors, liver. I have seen patients that have also breast neuroendocrine tumor, appendiceal neuroendocrine tumors, and we have seen an increased incidence of neuroendocrine tumors in the pediatric population. We can also see here on the graph on the left, a classification of the incidence of adult neuroendocrine tumors. And we can see here that there is an increased incidence of lung neuroendocrine tumors, as well as the small intestine neuroendocrine tumors and appendiceal neuroendocrine tumors. Neuroendocrine tumors can be classified into well-differentiated neuroendocrine tumors, as well as neuroendocrine carcinoma. Neuroendocrine carcinoma, the difference between the two of them is because neuroendocrine carcinoma is more rapidly growing, more aggressive. They can go to other places earlier compared to the well-differentiated neuroendocrine tumors that they have more indolent behavior. Something to discuss here, and I'm sure you have heard about it, is the Ki-67. Basically measures the percentage of tumor cells and is associated with tumor cell proliferation. This can also guide us to classify the neuroendocrine tumors into well-differentiated neuroendocrine tumors, as well as the more aggressive neuroendocrine carcinoma. Here in this picture, you can see the different type of neuroendocrine tumors, the grade of progression in different places, but also something that I want to mention here very briefly is that these neuroendocrine tumors, they also have a very distinct genomic presentation. However, we're still trying to understand what these markers means in terms of the behavior and in terms of treatments of neuroendocrine tumors. Throughout the years, we have some advancements in the treatment of neuroendocrine tumors. As you all know, most of you are familiar with the lanreotide or the long-acting octreotide or sandostatin, which has been very helpful in control of the tumor, but as well as Dr. Dan Lin discussed, there is evidence that these treatments can also slow down the progression of the disease. We also have some other treatments that we have used for neuroendocrine tumors, such as everolimus, and also temozolomide. Now in 2017, it was approved the PRRT with Lutathera or peptide receptor radionuclide therapy with Lutathera, and we're going to discuss briefly about it. And this treatment, we know that is efficacious for the treatment of neuroendocrine tumors. However, we're going to discuss later today that even though we know that this treatment is efficacious for neuroendocrine tumors, we still need to make this treatment more effective in shrinking the tumors as well. As we all know, some of you are very familiar with telotrestat. Another name for this is Armello. This medication blocks the serotonin production, and we know that this medication also helps for the treatment of the diarrhea. So even though throughout the years we have seen the some advancements of in the treatments of advanced neuroendocrine tumors, we still have some challenges in terms of management. We have very few FDA-approved therapies for neuroendocrine tumors, but the question here is when and what combination therapeutic approach should be used. Sometimes we know that depending of the amount of disease or how the tumor is behaving, whether it's rapidly growing or slow growing, we need to decide what will be the best treatment approach at the time of the presentation. But we still don't have standardized guidelines in the sense of when we need, what treatment we need to use and for how long the treatment needs to be continued. Another question that we have is like, what's a group of patients a particular treatment option should be used on? So we know that a lot of the treatments that has been used for neuroendocrine tumors, it focus mainly on the pancreatic neuroendocrine tumor, the small bowel neuroendocrine tumors, but how about the other sides of disease? Can we extrapolate the same information? And that's something that we need to do more research about it to understand which patient would benefit from which type of treatment. Now, neuroendocrine carcinoma, which is another neuroendocrine tumor which is more aggressive, we know that immunotherapy has a role in the treatment of neuroendocrine carcinoma. However, we still need to understand more about it to really determine which patients benefit from this treatment. However, there are other challenges that the patients face. Patients with neuroendocrine tumors as well as their families face unique challenges. And one of the reasons is because sometimes it can take long time to diagnosis and is often identified in advanced stages. By the time the patients goes to see the doctors, they already had advanced disease, the neuroendocrine tumor has spread to other places. Most medical centers has limited experience with these tumors, so patients often have to search for experts, and sometimes they have to travel outside of the home origin. So that can definitely generate a lot of anxiety and a lot of frustration going to other places where we don't know if the insurance will cover for that visit and so forth. Another frustration and challenges too is that sometimes we don't have a standardized treatment guidance, and that creates uncertainty and search for answers. And I see this all the time, that my patients can often feel isolated. And that's why, what will be a solution for this? A solution will be to develop an easily accessible national and international patient engagement network for NET research. And here, that's where organizations such as like the ENETs, the LACNETS can play a very important role to educate patients, as well as to establish a network of research that can help patients as well as researchers. And also patient care with advocacy and support groups is very important, such as the LACNETS, LACNETS has been doing an amazing job in educate patients and inform patients with neuroendocrine tumors. Other ones will be the Neuroendocrine Tumor Research Foundations, the NORCAL. There are different neuroendocrine tumors that are around the United States that can be a support to you. And that can definitely address some of the patient needs, as we discussed above, but they can also increase the access to patients for research. But also the NET researchers also faces hurdles to develop cures, because sometimes considering these neuroendocrine tumors are considered rare type of tumors, with these rare tumors, it can take a long time to accrue patients in clinical trials. Sometimes multidisciplinary care, or often multidisciplinary care, are needed for patients living with neuroendocrine tumors. We need to have your nuclear medicine physician, your oncologist, your endocrinologist, your radiation oncologist. We need to all be together working as a multidisciplinary team in order to give the best treatment approach for patients living with neuroendocrine tumors. But also at the same time, neuroendocrine tumors also are associated with cancer predisposition syndromes such as von Hippel-Lindau. Von Hippel-Lindau, besides neuroendocrine tumors, they are associated with pheochromocytoma, with hemangioblastoma of the central nervous system. And you need to have a multidisciplinary team that will definitely look out for all of the clinical manifestation and give you the best care. So this is very important to have a multidisciplinary care, because this is what is needed for the care of patients living with neuroendocrine tumors. But sometimes having this approach can also form silos in terms of lack of coordination and communication between teams, and that can also limit efficiency into clinical trials. We also understand there are limited diversity in terms of neuroendocrine tumor cell lines, meaning that we don't have enough preclinical models to do drug testing to then develop more effective treatments for neuroendocrine tumors. And also, fundraising is very challenging for this rare disease. And for all rare diseases, it's very difficult to do fundraising, less sources of grant funding. And that's one of the hurdles that we as a NET researchers also experiencing as well. One of the solutions to this is to have a more collaborative, well-funded research network for neuroendocrine tumors that could also promote development of new therapies and a standard of care treatment for patient care. And this is very important. I think in regards to the treatment of neuroendocrine tumors, we definitely need to have a collaborative approach with different institutions as well with different organizations to give the best care of patients living with neuroendocrine tumors. We're working very hard for the understand more about the biology of neuroendocrine tumors. And after what I discussed to you, one of the solutions could then have a more standardized cohort of patients that we can follow over time. And for that, we had developed the natural history study of children and adults with neuroendocrine neoplasm. This is part of the natural history study for children and adults with red solid tumors. The goal of the study is that we wanted to characterize the natural history of neuroendocrine tumors. All of the data that we're collecting is demographics. Also, we're collecting clinical characteristics, patterns of disease progression, response of or lack of response to therapeutic interventions, as well as disease recurrence and overall survival, how long these patients are living with this disease. We wanted to understand more about the biology and the clinical presentations of these tumors to better give the best care. And we feel that by having this natural history study can definitely help us achieve those goals. The natural history study will allow us to study you long term. We can also look back what has been done, and we can also follow you what can be done in the future. Patients that are enrolled on this study can be enrolled remotely in the sense that patients doesn't need to come to the NIH, or they can also come to the NIH, and then we can provide different assessment in terms of other research evaluation that we wanted to do to understand more about the biology of these tumors. We also, besides the patients that we wanted to study, we also wanted to understand more about the relative, if there is any germline or cancer predisposition mutations within the family that can help us understand about neuroendocrine tumors. We are doing a very extensive clinical and laboratory evaluations because we wanted to understand if there is anything abnormal that can be found in the lab that can help us understand more about how these neuroendocrine tumors behave. We're also doing a very extensive patient-reported outcomes in terms of the psychological and social functioning, something that we all know with neuroendocrine tumors, especially patients with carcinoid that produces a high level of hormones, they have profound diarrhea, and sometimes that can be quite debilitating, losing hours of work, and it can be very also frustrated for the patient that they cannot live a normal life. We wanted to understand how that affects every aspect of the patient, from the socioeconomic levels, from the psychological levels. So we wanted to understand how we can help patients in that aspect. We're also doing a very extensive molecular profiling on the tumor tissue, blood, saliva, and stool. And we are going to do a specimen repository in the sense that if we in the future found something that is interesting that can help us understand about the biology of these tumors, we'll go back and then do more assessments on those tumors. The whole goal of doing this is that we wanted to develop new therapies. We wanted to also understand how we can make better clinical trials, maybe with less patients that can be included, that can be of benefit of patients with neuroendocrine tumors. And we also wanted to establish screening guidelines for those patients. So the way that our natural history study works is that either the patient itself or the referring physician or the referring site we refer the patient to us. If patients initially will be consented to the main natural history study for rare tumors, we're going to collect all the clinical information, pathology, imaging reports. We discuss all of the findings, doing a multidisciplinary review, and provide an opinion to the patient as well as the referring physician. We're also doing in the tumor a very extensive molecular and genetic analysis, and that information will be shared with the patient as well with the physicians. Once we decide that this patient is eligible for our study, they can either follow on this study long term. We can then enroll them on the neuroendocrine tumor sub-protocol. We have a very specific protocol to study neuroendocrine tumors for pediatrics and adults. They can also be possible enroll on a treatment trial if available at the NIH. And those are all the possibilities of patients that are being enrolled on this natural history study. So what data we're collecting, and I have to say that has to recognize Dr. Riley and Dr. Wideman, who are the director of the Rare Tumor Initiative, for which I'm able and fortunate to collaborate and work with them. But the data that we're collecting is either for all participants that are unable to come to the NIH, that we're enrolling remotely. We're able to collect medical and family history. We're also able to collect patient-related outcomes such as pain, fatigue, depression, cognitive functioning. We also wanted to understand about the cancer characteristics by imaging, by pathology, by tumor specimen, and doing a whole exome sequence on the tumor as well. Patients that come to the clinical center, we're doing a more extensive clinical profile with very detailed history and physical examination. We may do imaging at the NIH. It's possible that we may do a biopsy if necessary to understand more about the molecular findings of the tumor. We're also doing a very extensive tumor immune profile as well as in the blood and in the tumor to understand of these patients who they may benefit from immunotherapy. By doing that, we also trying to establish cell lines and as well preclinical models when we're able to get tumors that, well, once we're able to collect that information, we should be able to then do more drug testing that can be of benefit for patients with neuroendocrine tumors. We have a wonderful genetic counseling that is very familiar with all different manifestations related to neuroendocrine tumors. And as we discussed, we have also very specific patient-related outcome. All participants that are eligible for our study, they also can be eligible for clinical trials at the clinical center. And also we can provide opinion for other studies outside of the NIH. So this is just very briefly about the type of biospecimens collection that we are collecting as part of our study. So for patients that are unable to come to the NIH, what we do is that we send a saliva kit. We're able to extract DNA. And with that DNA, we should able to do a germline testing. And then we compare that with the tumor. So we're doing a whole exome sequence on the tumor as well as on the DNA from the saliva to understand if there is any other cancer predisposition syndromes associated with neuroendocrine tumors. We also, what we're doing for patients that come to the NIH is that we're also doing a very extensive blood collection in forms of the, to study more about the immune microenvironment related to neuroendocrine tumors. We're also collecting more of the DNA as well to do further studies in the future. So what are the objectives of this study? So the natural history study will definitely help us to establish a more formal mechanism for patient referral. Also, we can also allow us to do a systematic collection of epidemiologic data and as well to collect tumor samples to do further analysis. The whole idea of having this study is that we wanted to understand and classify patients with neuroendocrine tumors from the clinical state, from the genomic, epigenetic alterations, as well as gene expression profile that could serve to better define the biology of these neuroendocrine tumors. And the whole idea is that with this, we should be able to correlate with clinical outcomes and be able to develop effective therapies. So we also wanted to address unmet need to also evaluate children and adults throughout the whole time of their disease course, because we wanted to understand the natural history, the clinical features, how these tumors behave over time. Do they change from more indolent to more aggressive behavior? Those are the questions that we have, and we're hoping that with this study we should be able to answer those questions. We wanted to also allow the identification of cancer predisposition syndrome, maybe some families are at risk to develop neuroendocrine tumors. We wanted to understand that. We wanted to do cancer screening for cancer prevention, as well to the risk reduction of neuroendocrine tumors. This is our objective of doing the studies, and this will be a longitudinal study, and we hope that we can answer all these questions by the end doing the studies. And as we discussed earlier, I think it's important to also understand the patient-related outcomes, how having this disease is affecting you from the socioeconomic level. It's the cause fatigue, the diarrhea, how does it affect you in your personal life, in your work life? I think those are questions that are important to answer as well. I'm going to discuss now the current clinical trials at the National Institute of Health for Neuroendocrine Tumors. So the first one that I'm going to discuss is a study that we have for pheochromocytomas and paragangliomas. As I said earlier, pheochromocytomas and paragangliomas is also considered an orphan disease. It's very rare. It's a tumor that is localized in the adrenal gland. The adrenal glands are on top of the kidney. They have also very specific clinical profile in the sense that they're able to produce adrenaline. Catecholamines or adrenaline. So as you can imagine, when you think of adrenaline, that's what is caused your blood pressure to go high, your heart rate to go up. So there are tumors that can cause this excess of adrenaline that can be quite symptomatic from their disease. So that's a very rare disease that has also very limited treatment options as well. And another thing important to know about these tumors is that they also express somatostatin receptors. The majority of these tumors express somatostatin receptors very similarly to neuroendocrine tumors as well. So one thing that we wanted to do and understand more about finding more efficacious therapy for neuroendocrine tumors is that if we can do something that could stop the DNA repair. So as we know, and on this picture, whenever there is either a stress or any type of treatments that breaks this DNA, because the DNA, everybody has their DNA, it goes into a certain way, but whenever we give a treatment that causes a nick on that can cause damage of the DNA, there are certain enzymes. We call that the PARP enzymes. And I'm sorry that I'm getting a little bit too technical here, but the name of that PARP enzyme is also called polyribose polymerase. And basically, what it's doing is that once that happens, the cell is trying to repair itself. And that's why by using what we call these PARP inhibitors, as we can see here, there's 3 different type PARP inhibitors, such as olaparib, rucaparib, niraparib. Those are the ones that has been approved for other cancers, such as ovarian cancer, prostate cancer. But when you use one of these medications or this drug, what it is blocking that repair, and is to put in what it's doing is this action is lethal to the cell and kill the cells. So that's basically the rationale, and that's why we nowadays, in terms of finding other treatment options for different tumor types, we're trying to combine treatments with these PARP inhibitors to make the treatment more efficacious, to stop for the cells to trying to repair itself. As we can see here on this picture with the survivals, and I know Dr. Danlin explained to you very nicely about the clinical trials and to understand what is Kaplan-Meier are for, but basically this is what is telling me what is the survival of the cells, how long the cells are able to survive on this graph. And basically here what I'm trying to say is that the combination of temozolomide, who has been a drug that has been used for neuroendocrine tumors, with olaparib can definitely help the cells to survive even longer. And this is the same rationale that we're using with different tumors, but this is the rationale that we're using here for the treatment of pheochromocytomas and paragangliomas. And because of that, we now have a trial that is going to be open very soon at the NIH. That is the combination of temozolomide and olaparib versus temozolomide alone. This trial is in collaboration with Dr. Kim Perez, who she's at Dana-Farber, and we're going to include all patients that had pheochromocytomas and paragangliomas, either advanced or unresectable pheochromocytomas and paragangliomas. And what we wanted to know by using this combination, either combination or temozolomide alone, is that we wanted to know if by using this medication, does these patients live longer with their disease? Do we see does the tumor shrink? We also wanted to understand with this combination if there is any more toxicity related to this versus the temozolomide alone. So those are the questions that we want to answer with this trial, and is going to be available very soon at the clinical center as well as other institutions within the United States. So we discussed earlier about the importance of doing a gallium scan in patients with pheochromocytomas and paragangliomas. So we also discussed, very similarly to the pancreas neuroendocrine tumors or the small bowel neuroendocrine tumors or even the lung neuroendocrine tumors, that they express somatostatin receptors. And it's the same with pheochromocytomas and paragangliomas. So we can see here and on your left side a picture of the FDG PET scan, and we can see tumors that we can see on the picture, but when we see on the gallium scan, you can see every single tumor related to pheopara. So the tumors are very well delineated. We can see how the gallium scan can pick up other tumors that were unable to be seen on the other imaging studies. So this definitely type of gallium scan has reformed the way we use diagnostics for different type of neuroendocrine tumors, and the same with pheochromocytomas and paragangliomas. So, and this is just very briefly to discuss what are the basics of theranostic. What is theranostics? I'm sure you have heard this throughout the day today for the treatment of neuroendocrine tumors, but basically theranostic is the combination of the term therapeutics, treatment, and diagnostic, how we're able to diagnose or localize the disease. The same thing with the gallium scan. So we have a diagnostic modality, which is the gallium scan, which binds the somatostatin receptor. Whenever you inject that type of radiation as a diagnostic tool, it binds to the somatostatin receptor, for which we're able to see these tumors. If they express the receptor, we're able to see it on the scan. And at the same time, we have the treatment modality that have the same rationale as well, that finds the somatostatin receptor, which is found on the surface of the tumor cell. So peptide Lutathera is a type peptide receptor radionuclide therapy, and it's a type of radiation that can be given to treat advanced disease or metastatic disease related to neuroendocrine tumors. It is approved by the FDA for the treatment of pancreas neuroendocrine tumors as well as for the GI neuroendocrine tumors. It is not approved for the lung neuroendocrine tumors. However, research is now ongoing in order to determine the benefit to get it approved so patients can get Lutathera with lung neuroendocrine tumors. But the same for pheochromocytomas and paragangliomas is not approved, but we have a study at the NIH that we wanted to understand more about what is the benefit of using Lutathera in patients with pheochromocytomas and paragangliomas. So this is a phase 2 study. So Dr. Dantlin explained to you the difference between this clinical trial and clinical trial development. We're running this study together with Dr. Franklin for nuclear medicine at the NIH. We wanted to understand is by giving this treatment, do those patients live longer with this disease? Can we see can the tumor shrink by giving this disease? Can we improve the quality of life? Because as we discussed earlier, these tumors with pheochromocytomas and paragangliomas, they produce excess of adrenaline, and that causes high blood pressure, palpitations, sweating. By giving this treatment, can they improve the quality of life? So those are the questions that we have by giving this treatment. We're still accruing those patients actively, and that is available at the NIH and open to all patients. I'm sure you're familiar with this study. Throughout the day, I'm sure you have heard about about the NETHER study and the Lutathera for neuroendocrine tumors, and we know that this treatment works. This was the study that was led by Dr. Jonathan Strathford that was published in 2017. And basically, the study is showing you that there is benefit of using Lutathera in patients with neuroendocrine tumors. We see that there is benefit of patients that they can live longer with the disease, but also they stabilize the disease for longer time. So this is very important. This has reformed the way we're seeing now neuroendocrine tumors. However, something to keep in mind here is that even though we know that it works in neuroendocrine tumors and it's a very good treatment for neuroendocrine tumors, the response rate is not as high. So now the ongoing research is to make Lutathera more effective. And that's why, the answer is, how can we do that? Can we combine Lutathera with something else that make the treatment more effective? And that's the question that we have. So Lutathera, as I discussed earlier, is a type of radiation. Because it's a type of radiation, it can cause DNA damage to the cells. As I discussed earlier with another study, sometimes when there is DNA damage, the cell is going to start to repair itself with this enzyme that is called PARP enzymes. And the question is, does it make sense to give drugs such as olaparib that blocks that enzyme in order to doesn't let it repair and then cause cell death? And that's exactly the question that we have here. So we know that the type of radiation does cause breaks on the DNA, and there is a potential synergy, meaning that can be more active when you combine with the olaparib. And that's what is and that's where I want to show you here in this picture. And this preclinical study done in neuroendocrine tumor cell lines, when we can see on the first, and if we can focus right now with the blue cells and the pink dots, so we can see the first square is the neuroendocrine tumor cells. But when we add the Lutathera, we can see that the pink dots are the DNA breaks. We can see with the Lutathera, it caused some DNA break. So now the third one is only olaparib alone, or the PARP inhibitor alone, and we can see some DNA break. However, when we give it together, we can see more pink dots. So it means that there is more DNA breaks. And that's the rationale that led us to then have this trial of Lutathera in combination with olaparib. It's a first-in-human study. So that means this combination has been used in human. So we need to do a phase 1 study to determine what is the safe dose. We have a dose escalation with olaparib. The Lutathera is a standing dose. We don't do any dose reduction of the Lutathera. It's a standing dose that has been approved for all neuroendocrine tumors. And then we add olaparib and escalation dose, starting with a low dose, and then we go a little bit higher to determine what is the safety and tolerability of this combination. Once we know what is the safest dose to continue with a phase 2 study that is going to be mainly specific with neuroendocrine tumors, and is going to be a fixed dose of the Lutathera and as well as the olaparib, then we wanted to understand, is this more efficacious? Is this combination helps to shrink the tumor? If this combination helps for the tumors to get stabilized and to live longer with this disease? And that's the question that we want to answer with this study. I wanted to also mention here that it's important, something that I always do with my patients, I want to educate it about what other treatment options are out there, what clinical trials are available for patients with neuroendocrine tumors. And that's why here it's important that you're familiarize yourselves with clinicaltrials.gov, because it's important to know that there is any other treatment options besides the ones that my doctor is giving me. And as you can see here, it's important that you type the condition, in this case neuroendocrine tumors. You can also type the place where, if you want to find a place closer to you. We can also type if there is specific name drug or investigator that you want to add, and you can plug all that information into this website, so that way you can find other treatment options available for neuroendocrine tumors. And here there are only a few that I was able, when I went to the clinicaltrials.gov, there is a few here that I would like to mention. There is the cabozantinib study, Jennifer Chen at Dana-Farber leading this study. We also have the NETHER study, the Lutathera for higher grade neuroendocrine tumors. Dr. Jonathan Strathberg is leading that trial. The same with the tamoxifen for well-differentiated neuroendocrine tumors. There is a rationale that these neuroendocrine tumors, they may have a hormone receptor, estrogen receptor in the surface of the cells, and maybe that's the rationale why tamoxifen can be used in those patients. It's still in the setting of a clinical trial. We have another trial with the tripine, the same rationale with the olaparib. We wanted to make this treatment more efficacious. Dr. Aman Chauhan is the investigator on this trial. And then we have also the combination of telotristat, another name for this one is Remelo, in combination with Lutathera for neuroendocrine tumors. All of these are available, and there is always an information on who to contact in order to determine if you're eligible for any of these studies. Lastly, I would like to mention in the last two slides in terms of what is the research ongoing with high-grade neuroendocrine tumors. As I discussed earlier, the neuroendocrine carcinomas are the ones that are more aggressive, more rapidly growing. Those are the ones that can metastasize earlier and go to other places, and that's why it's important that this is an unmet need, that we need to determine more effective treatments for this type of neuroendocrine tumor. At the NIH, I'm working very closely with Dr. Thomas. Dr. Thomas is the one who is developing treatment trials for small cell lung cancer, which is another type of neuroendocrine tumors. And we're trying to open a trial that is specifically for high-grade neuroendocrine carcinoma. This is a combination of two drugs. The first one is lurbinectedin. So lurbinectedin is a drug that basically damages the DNA. In combination with berzosertib, what it does is inhibit the ability of cells to divide, and it even causes more DNA damage. So, and there is already studies that show that the combination of the two can be more efficacious for the high-grade neuroendocrine carcinoma. So this is a first-in-human study because this combination has not been used before. So we're trying to determine the best dose to then move to the more phase 2 specific for neuroendocrine tumors, and that's one of the treatment trials that I'm working very closely with Dr. Thomas. And it should be open in the next month or 2 months, and it will be available for small cell as well as for high-grade neuroendocrine carcinomas. The same thing if there is a patient that has a higher grade neuroendocrine tumors or neuroendocrine carcinoma. Also, clinicaltrials.gov has other treatment options that it will be worthwhile to explore. Also, one of the other treatment options that we're working very closely is also given the combination chemotherapy with other drugs to determine if we can make this treatment more efficacious. Immunotherapy, that's another thing that we're working very closely as well to determine of the patients that understanding the tumor immune microenvironment and to determine which patients may benefit from immunotherapy. But those are the treatment options that are available at clinicaltrials.gov, and all this information will be given to you, so it's available to you at any time. So lastly, I would like to recognize everybody that I'm working with. This is a teamwork. I'm not doing this by myself. I'm working with very exceptional investigators at the NIH. I have investigators that focusing on the tumor immune microenvironment. Dr. Polina is one of them. I have an investigator who's trying to understand more about the tumor biology of any extrapulmonary, more aggressive neuroendocrine tumors. I have an investigator who's only focusing on preclinical models, Dr. Kumar, and he has been quite successful in developing some of these models for neuroendocrine tumors. Dr. Lin is a nuclear medicine and oncologist who's also working on the theranostic program at the NIH. We have other investigators who has very specific interests on different type of neuroendocrine tumors, and I feel that this is the only way that we can advance the science forward, that we work as a team. And we always put all our minds together to definitely excel and advance the science. So I have to acknowledge everyone that I have worked with. This is just a few of them that I have been fortunate to work with at the clinical center from different departments at the NIH, from surgery branch to radiology department, from nuclear medicine, from endocrinology, surgery, pediatric oncology, the developmental therapeutic branch, which is my current home. I think that I have to acknowledge everyone that I have been working with, but more importantly, I have to acknowledge all of the patients, because without them, none of the studies will be possible. So I have to end with these slides. I want to honor our great friend and the founder of LACNETS. As you all remember, Giovanna Invesi was the heart of this organization. I was very fortunate and honored to meet her last year at the ENETS meeting. We have this cooking class, and you can see how happy she enjoyed to be with all of us. She really cared about each one of you. She really wants you to fight for this disease, to find better treatments. She did all this for you, and I have to thank her for that. There is a quote by Albert Pike that I want to say to all of you. Whatever you do for you, stay with you. Whatever you do for others, and the world will be always immortal. and she wants you to fight every day. She wants you to work together, and she wants you to find better treatments. And that's what we're here for. Without you, without her, we wouldn't be where we are, and we're grateful to you all. Again, this is a team effort. So individually, we are one drop. Together, we're an ocean, and we can do this together. Thank you all.